Hi there, my name is Christopher Situ.
And my name is Jason Yee, and we're both grade 11 students from Oatscona Academic in Edmonton, Alberta.
Our investigation centers upon a disease-causing protein known as the Proteinaceous infectious particle. Healthy prion proteins are found in a variety of animals, including humans, and although their full role isn't entirely understood, they have been observed to participate in various cell activities. These include neurogenesis, hormonal homeostasis, cell signaling, and stress protection.
However, when a prion protein misfolds into the disease-causing Scrapie's form, it becomes capable of inducing changes in other healthy proteins, turning them into prions as well. If given enough time, the subsequent protein amyloid aggregates can build up in the brain and severely hinder brain activity, leading to death.
A notable example of prion disease is none other than mad cow disease. It spread in the UK in 1993, for example, caused the deaths of over 180,000 cows and 177 people. Even closer to home, our home province of Alberta had its own brush with the disease in 2003, which had devastating economic implications on our economy, especially as global markets like the US and the EU placed trade embargoes on our bees. As such, there are benefits beyond therapy to researching and combating prions. With that said, what exactly was our project and how did it work? Well, we utilized the program Chimera to conduct the simulations of drug docking on prion proteins. Here, in fact, you can actually see the surface depiction of the Scrapie human prion protein, as well as the docking sites of our drugs of interest. Our research was backed up by online publications as well as our mentor, Dr. Luda Darash. But additionally, Professor Holger Riel and Professor Maria Stepanova of the University of Alberta, alongside Miss Loretta Ng, monitored our progress and kept us on the right track.
In fact, our entire investigation was inspired by our mentor, who was doing studies on prions before. We saw this as an opportunity to use our knowledge and research to contribute to a discovery that could possibly save a lot of lives in the future, as romantic as it sounds. Anyways, using the program Chimera and the extension AutoDock Vina, we conducted simulations for the binding of compounds onto the disease-prone mutant forms of the Human Prion Protein. As a quick remark, here you can see the ribbon representations of the same Scrapie's protein and docked compounds. And out of these 119 tested compounds, 9 were selected for further analysis due to their remarkably high binding affinity.
By our results, we eventually concluded that charged compounds bind better than their uncharged counterparts, and that compounds bound generally best to the Scrapie model and worse to the experimental mutant model.
These compounds were observed to bind in 5 key sites across the 3 proteins. These docking locations and newfound drugs of interest take strides towards the progression of prion disease therapies and the understanding of cellular prion function, which are still a mystery to us.
Now, this begs a very relevant question. How could we continue this project? If given more time and resources, we could easily extend the project by simply finding more compounds to simulate and analyze with the same procedure. However, this methodology would really only work as long as we find compounds with evidence to be of use. In fact, Professor Holger Riel recommended we find specific justification, for example, in research papers, before testing in order to ground our investigations in reality.
Our ambitious idea would be taking our results and developing an entirely new treatment for prions based on the best binding compounds and residues they're bound to. That being said, this investigation wasn't all sunshines and rainbows. We encountered various problems along the way, and just a few were kept as visual records, as shown here.
As evidenced by our lab journal, there were several times where we had to change or completely revise our procedure. The trial and error process forced us to problem solve and innovate to overcome our challenges. Consequently, in my opinion, this both enhanced the final product and more acquainted us with the research process.
Furthermore, the dozens of hours spent simulating, resimulating, and analyzing were very exhausting. However, simply knowing that something beneficial could transpire out of our efforts and small victories we achieved along the way pushed us forwards.
Moreover, the open environment where we were free to explore different areas of biotechnology made the entire investigation much more engaging. This allowed us to choose something that we were actually passionate about and gave us the support and resources necessary to make something out of it.
The large time frame for this project also allowed us to experiment with various techniques, tools, and scientific methodologies in our investigations, and ultimately to fail but try again. We made a fair bit of mistakes, but through the trial and error, we were able to learn and grow. And with that, that concludes our video presentation. Thank you so much for watching. We hope you enjoy. And coming out, you may now share some of our enthusiasm for this project, this topic, and maybe where it's headed next.
Who knows? One day in the near future, prions could be nothing more than a vestigial remnant of the past, and we hope that our investigation can contribute to this vision.